Hi, my name is Nicolle. I'm a PhD student at PUC-Rio, and this work was developed with Professor Márcio Carvalho, and it's entitled Lamellae Generation and Dynamics During Gas Invasion of a Porous Medium Occupied by a Surfactant Solution. Water and gas are commonly injected in the reservoir to maintain the pressure and displace the oil towards production wells. And the amount of oil displaced is both a function of how well the reservoir is swept by the injected fluid and how well the oil is displaced in the pore scale, the residual oil saturation. The enhanced oil recovery methods are the applied techniques to maximize oil production through the interaction between the injected fluid and the rock oil system. Each EOR method acts in one or ideally both of these two factors. Different methods can be used with their own advantages and disadvantages. One of them is the gas flooding. When we inject gas, the gas flows through the upper part of the reservoir, leaving a large amount of oil behind. This is called early breakthrough. Also, we have the formation of these instabilities called viscous fingering. So the idea here is to reduce the mobility of the displacing phase. And we can do that by alternating water and gas. We have a better sweep, but we still have oil remaining in the reservoir. In the third case, in the surfactant alternating gas injection, first we inject a slug of surfactant solution, then we inject gas. In this case, foam is formed inside the reservoir, and foams are better injection fluids than aqueous solutions because they are less sensitive to gravity and permeability heterogeneities. And also foam provides a more uniform sweep front here. It's a potential solution to mitigate the adverse effects of gravity override, early breakthrough, reservoir heterogeneity, and viscous fingering on the efficiency of gas injection. Foam behaves in different ways in a porous media. We can have a continuous gas path. We can have bubbles flowing. This is called a bubble train. And we can have trapped gas here in bubbles. Foam has high apparent viscosity by trapping gas in bubbles, reducing the flowing fraction of gas, and providing capillary resistance. So the idea is to decrease the mobility ratio by decreasing the relative permeability of the displacing phase and increasing the viscosity. The idea of this work is to study foam formation during drainage process in a 2D porous medium micromodel by visualizing the pore scale displacement, and obtain pore scale information that can be used to improve continuous model of foam formation and flow in porous media. To do that, we're going to use a 2D micromodel made with glass by Microneat. Here is the picture of the micromodel. We have the glass pore structure here, two buffer zones, one in the entrance and the other one in the outlet. Here are the sizes of the micromodel, their properties, the pore volume, porosity, and permeability, the sizes of the channels. The fluid used here is a mixture of water and sodium udaldecyl sulfate. And the surface tension of the surfactant solution and gas is 34.4 millinewton per meter when we are above the CMC, the critical micelle concentration. The experimental setup used consists in a syringe pump, a gas-tight syringe, a pressure transducer to monitor the pressure, the micromodel, an inverted microscope, and a computer to do the image processing. First, we saturate the device with a surfactant solution at different concentrations. Then we inject gas at a constant volumetric flow rate. It's 1 milliliter per hour. Then we measure the pressure and visualize the flow dynamics. Here we can see the gas invading the micromodel from the left to the right. The device was completely saturated with surfactant solution, dyed in red color. Also, you can see the formation of distinct liquid films. They're called lamellae. During time, we have a decrease in the liquid fraction. So in the beginning of the experiment, we have a higher liquid fraction, and the bubbles have this shape, these circular shapes here. And at the end of the experiment, we have a lower amount of liquid. Here, it's less than 1% of liquid. We have these structures called diaphragm. The bubbles have these polyhedral shapes with curved edges. Here is the saturation profile after the injection. The device is completely saturated with gas, and we only have liquid present in lamella form. Here we can see the percolated gas path dyed in red. Now analyzing the effect of concentration, here we have two cases with different concentrations, but as we are above the CMC, we have the same value for surface tension, but completely different behavior in the pressure. So when we see that this red curve is the higher concentration case and this blue curve is the lower concentration case, you can see that the maximum pressure is higher for the higher concentration, and also the time that takes for the gas to find a percolated gas path, it's 100 minutes comparing with 70 minutes for the lower concentration case. When we are comparing with a case with no surfactant, here this black curve, we can see that it only takes 8 minutes for the gas to find a percolated path, and the maximum pressure is much lower than the other cases. We can conclude that the surfactant solution completely changed the two-phase flow in a porous media. So analyzing the pressure behavior for different concentrations, we can see that as we increase the concentration, we also have higher values for maximum pressure. And the pressure increases during the test. And when the gas finds a percolated path and we're not forming lamellae anymore, we have a pressure drop and the pressure stabilizes in these plateaus. Here we have the formation of two plateaus. We don't know yet why we are forming these two different plateaus, but to understand the dynamics of the whole process, we are starting to visualize the flow in a region of the micromodel. This yellow square here shows the region that we are analyzing now. We can see here a video of the gas flowing, foam formation mechanisms, and quantify the lamellae and its velocities. To do that, we're going to use an ImageJ processing tool. The first input is an image of the device completely saturated with air. This is our matrix. We're going to use the binary image, only black and white. The second input is an image with the lamellae filled with, the device filled with lamellae. We also use the binary image. When we subtract this image from this one, we can identify the lamellae and quantify. We can see their positions and their velocities. And calculating the lamellae number in millimeters square as a function of time, we can see here the plot for different concentrations. When we have the higher concentration, we also have the higher number of lamellae. And for the lower concentration, we have lower values for the lamellae. But when we achieve the steady state condition, we have almost the same values. So when stabilized, the number of lamellae are almost the same. So my conclusions are that the gas foam films, they are formed, create a resistance against the gas flow, and increase the magnitude of viscous forces against capillary forces. The pressure behavior do not depend only on the surface tension. The surfactant concentration changes the population of lamellae that compose foam, impeding the free motion of gas through the porous medium. We are obtaining detailed information on the dynamics of lamellae formation and speed that can be related to the microscopic flow behavior. Here, it's another analysis that we're doing now. We are tracking the lamellae. We can see the gas invading here. The lamellae are these white parts, We're using image 2 processing tool, image J processing tool. And the data that we have, it's this, it shown in these images. Here is the mean velocity of the track. We can identify in these blue regions where the gas is trapped and where the gas is flowing. It's this region, in this track, the gas is flowing, the Lamellae, they are flowing, and here the gas is trapped. Another ongoing work is a three-phase flow. So first we saturate the device with an oil phase. Here the oil phase is dyed in brown. Then we displace the oil using an aqueous solution. In the first case is water, so no surfactant is added here. In the second case, we added surfactant, the SDS, 6.2 grams per liter. Then we inject gas. So in the first case, we can see that the gas flows through almost the same path as water, and the residual oil saturation value is a little bit lower. But when we inject gas after surfactant solution, we have foam formation. And this foam formation leads to a lower residual oil saturation. You can see the foam here in detail. I would like to thank you and thank all the founding agencies.